The third and last source of genetic variation, so big and bold in your notes, lots of highlights, big stars. This last source of genetic variation is called independent assortment. Let's talk about what independent assortment means. So we're going to have our homologous chromosomes line up along the equatorial plane. So we're going to play a little game. I have a coin. I'm going to flip my coin. If I get heads, I'm going to put the green chromosome on the left side, and if I get tails, then I'm going to put the green chromosome on the right side. So now I can go ahead and draw in my blue chromosomes.
We can use a diploid or two-end cell with just two pairs of chromosomes to illustrate a genetics concept called independent assortment. We will track the movement during meiosis of the cell's two pairs of homologous chromosomes. Before meiosis begins, the cell synthesizes new DNA and thereby replicates each chromosome. Early in meiosis, the pairs associate. Note that chromosomes in a pair are not quite identical. In one pair, for example, one chromosome carries a recessive allele of a gene, indicated little r, and the other carries a dominant allele, big R. The two pairs line up at the cell's midplane during metaphase 1 and may adopt one of two alternative alignments. The alignments provide the basis for independent assortment. The cell may adopt alignment 1, in which the little r and big Y alleles end up in the same daughter cell. Or the cell may adopt alignment 2, in which the little r and big Y alleles end up in separate daughter cells. From a large pool of cells that initiate meiosis, on average, half of them adopt alignment 1, and the other half adopt alignment 2. During the second half of meiosis, the two cells from each alignment divide into 4 haploid cells. The result is an equal number of the possible genotypes: little r big Y, big R little y, and big R big Y. Because there are equal numbers of genotypes, we can say that the alleles of the R and Y genes assort independently during meiosis. Note that alleles of different genes always assort independently if the genes reside on different chromosomes, but not necessarily if they reside on the same chromosome.